Eukaryotes have membrane-bound organelles, and they make up roughly 60 to 80% of their cell volume, comprising their internal structures. Now, I found that interesting, the 60 to 80%, because a lot of times when you read textbooks, they have everything super spread out. You think, okay, there's a ribosome over here, and then there's a bunch of space, and there's a nucleus, and then over there we have the Golgi. And in actuality, they're much more scrunched together, and it's much more full than we give it credit. Okay, so we're going to go through some of those internal structures. The first one is the nucleus. It's a compact sphere-shaped organelle. It's probably going to be the most prominent if you're looking at it on a microscope. The nucleus has a nuclear membrane. So as we mentioned before, we have the nuclear membrane, and it is going to be actually two sets of membranes. So you'll see here, there's one, and then a second one connected with nuclear pores. And this allows things to go in and out as needed. We also have the nucleolus. This will appear darker if you're doing microscopy. And this is a site for rRNA synthesis, or the ribosomal RNA. Chromatin is the term we use to describe the DNA and any proteins associated with it. So it could be the DNA with some of its replication factors, DNA with its histones, any of that that's any of that protein that is assisting the DNA. The endoplasmic reticulum is a series of membranes used to transport and also used in storage. It's going to be continuous with the nuclear membrane and comes in two varieties, the rough and smooth endoplasmic reticulum. These spaces in between those membrane folds are referred to as cisternae. And the rough ER is known as rough because it has, if we zoom in over here, it has ribosomes on the outside of its membrane. And you can see here, if we have the ribosomes on the outside, they can actually insert, as they're making the proteins, into the cisternae, into those spaces where it can get further processed. This is the site for lipid synthesis and hormone synthesis as well. The Golgi apparatus should be adjacent to the endoplasmic reticulum. So if you're looking spatially at the cell, you should have the nucleus. Right next to it should be the ER, and right adjacent to that should be the endoplasmic reticulum, the Golgi apparatus. The Golgi is the site where proteins are going to be modified, packaged, and sent to their final destination. So these ones are little pita bread sacks that are membrane sacs. They're not going to be continuous in the same sort of maze-like way that the endoplasmic reticulum is. Instead, it's going to be a series of, like, I think 7 pouches all smushed together. The endoplasmic reticulum will bud off. You can see here tiny purple membrane-bound packets of the proteins they've been working on, and they'll come to this side of the Golgi apparatus. The transitional vesicles will be picked up, and then they can be passaged down, kind of like, I don't know, a beach ball down the end here of the Golgi apparatus till they get to the final pouch where they should be properly modified and packaged into vesicles to be released or sent throughout the cell. The finished vesicles are often known as condensing vesicles. Okay, so if we put all of those together, this is nature's assembly line, where you start out with the DNA, and the DNA is used to make RNA, which then we send out here, and that RNA gets made into protein on the ribosomes, and they get put into little budded vesicles here in purple, where they get passed to the Golgi, where that protein's processed and packaged, and then it can be sent out to be secreted or exocytosed. Okay, so the lysosome is a vesicle that contains enzymes for digestion. So they'll have very specific enzymes to move a reaction forward or expedite it. It can be for the digestion of food. It could be for recycling of organelles or ribosomes. It could also be for digesting invasive organisms, like if it found a bacteria that was not supposed to be there. These lysosomes originate at the Golgi. There are specific types of lysosomes known as peroxisomes. So again, they will look very similar, vesicles with molecules within. These are just vesicles that have specialized enzymes for breaking down hydrogen peroxide. Phagocytosis is the process of cell eating or invaginating and taking things in. So if we look at the process of phagocytosis and breaking it down in the lysosome, you would start over here with the engulfment of food. So you have maybe a paramecium or something that senses a food particle outside. It will pull it in and form a vacuole or a phagosome. You'll see here where it kind of sends out protrusions of the membrane, pinches around the food particle. And now we have a food vacuole. Now the food vacuole is then going to fuse with these lysosomes down here. So now we have enzymes being released next to the food where it can be broken down, digested into smaller pieces so that it's usable by the cell. Vacuoles are used for storage. So they're membrane-bound organelles, like where you can store fluids. If you are like a plant, you might save water for later, or solid materials that can be digested, excreted, stored, any of the above. It's saved for later. These are often found in phagocytic cells or cells that can eat. So phagocytosis is cell eating, cells like white blood cells, that their whole job is to eat pathogens or protozoa. Food vacuoles can merge with the lysosomes, like we saw in the last slide, to help digest the molecules that have been taken in. Sometimes you'll also see vacuoles that are used to store foods like fats or glycogens that for long-term storage for when food is not so readily available. Some protozoa, or these single-celled freshwater organisms, will use them to help regulate osmotic pressure to keep the right amount of water inside to outside. The mitochondria is a membrane-bound organelle used for energy generation. It has two sets of membranes, an outer membrane and an inner membrane. The folds of its inner membrane, shown over here, these little lines, are known as the cristae. And these cristae membranes are the ones that hold the enzymes, the electron transport chain. It's going to help produce the ATP. Mitochondria contain 70S ribosomes, which are the same size as the ones that we saw when we talked about prokaryotes. So there are some hypotheses out there, or theories, that mitochondria were actually originated from prokaryotes. So this is the endosymbiotic theory, the idea that mitochondria were once prokaryotes on their own, and now they're just engulfed inside of another cell and are now reliant upon them. A lot of this idea comes from the 70S ribosome. Okay, so some more pictures here. We have our cristae shown here in pink, where we would see the electron transport chain. We have the matrix in between in light blue. We have our 70S ribosomes. It has its own genome. That's another reason people have floated the idea of the endosymbiotic theory, though it has a very, very minimal amount of DNA. Chloroplasts are the light harvesting organelles that would be found in algae or plants. We're not going to focus a lot on them because they're not in the majority of microscopic organisms, but it is important to note that you would find these membrane-bound organelles if it's a photosynthetic organism. Ribosomes are the sites for protein synthesis. So if we're going from RNA to protein or translation, this would be where you would be doing that. They're made up of rRNA or ribosomal RNA and protein. Same sort of idea as what we saw in chapter 3. They have two subunits, the large and small. In eukaryotes, though, they're going to be a little bit larger. Their sedimentation rate is 80S, so large subunits 60, small is 40, instead of the 70S ribosome that we saw in prokaryotes and in the mitochondria. The cytoskeleton is comprised of actin, microtubules, intermediate filaments that are going to be a crisscrossing flexible network of structural proteins. So you would find them right underneath around the nucleus. You would find them around the inside of the cell membrane. You'll find them crisscrossing throughout, providing kind of highways throughout the cell. You would also find them in the flagella to help with mobility. They are pretty much everywhere. The cytoskeleton functions to give structure and also helps with molecular transport. So back to Bio 1 with dynein and kinesin walking on the cytoskeleton to help move molecules from one side of the cell to the other. There are three main types of proteins that are comprised in the cytoskeleton. You have microtubules. These are going to be very long, hollow proteins that are used to maintain shape, transport molecules, and then you also see them in that kind of 9-2 arrangement of the flagella. Actin is going to be thinner proteins that are found throughout the cell. We think of them often when we talk about like muscles and muscle contractions. They're also going to be more concentrated around right underneath the cell membrane, helping form kind of that meshwork to give structure to the cell known as the cell cortex. And we have intermediate filaments. These are a structural reinforcement throughout the cell, and you'll find a lot of them next to the nuclear envelope, helping assist in providing that circular shape of the nucleus and its rigidity.